Right away. So welcome everyone. So today we are diving into single cell genomics. And we're very fortunate to have two guest lectures by Fabian Theiss and by Romain Lopez. So we're going to talk about why single cells, why traditional approaches, don't necessarily work, and how did we come up with the modern day single cell RNA sequencing. And we're going to talk about how single cell technologies evolved and giving us rise to today's technologies through three different main paths. And then we're going to talk about how we go beyond RNA with single cell ATAC-seq and multi-omics at single cell level. Then we're going to talk about how to deal with noise, doublets, and other single cell issues. Then we're going to classify the different computational challenges in single cell data analysis and talk about some emerging deep learning methods for single cell data. But then most of the lecture is going to be spent on our two guest lecturers, Fabian Theiss, who developed DCA, and Romain Lopez, who developed VC SCVI. So let's dive right in. So why single cell profiling? The reason is that individual cells are dramatically different from each other. Namely, if you look at cells, in the body or plated, they are extremely different. They're different because of their environmental stimuli. They're different because of their interactions. They're different because of the cell cycle phase that they're in, because of the transcriptional bursts that they're undergoing. So every cell is actually very different from each other. So when you're looking at bulk data, it's very difficult to get at the diversity of those cells. Cells are also different because of their differentiation trajectories. If you look at the hematopoietic stem cell lineage, it differentiates down very different paths, and there's almost a continuum of cellular identities as you go down that differentiation time course. Also, there's dramatic differences between individual cells, even within a particular given cell type. Signaling can differ dramatically because of, for example, the expression of the signaling receptors that every cell expresses on its surface, but also because of the gradients of signaling molecules and their accessibility to every individual cell. And if you look at the microscope at individual cells and how they're expressing particular receptors or particular factors or particular response elements, you see great variability. And lastly, in the case of cancer, you also have genotype differences between individual cancer cells. Basically, the tumor cells themselves undergo dramatic alterations in copy number, in large segmental duplications, or in individual genes, which then leads to the phenotypic diversity between these cells. So when you look at bulk samples, you will end up with some kind of average measurement that might actually not represent the population. The population of cells might have, either very high expression or very low expression for a particular gene. And the average might simply suggest that, well, this gene is expressed at intermediate levels. Or you might lose very rare events. Maybe one cell out of 10,000 expresses that RNA, but when it does express it, it expresses it to very large amounts. So these are the kinds of things that we want to capture with single cell data. And there's many different technologies that have preceded the current explosion in single cell data analysis. So some of them are basically using real-time probing, different types of amplifications. They have specific advantages and disadvantages. And these include imaging technologies or single cell PCR amplification, in situ sequencing that allows you to directly sequence RNA molecules from a field of imaging to see where are those individual RNA molecules spaced across many cells. But then each of those has very different limitations. So the foundational technology underlying all of these is this ability to amplify individual RNAs and to then capture those RNAs from extremely small samples. In contrast to DNA, where you only have one molecule per time, RNA has many molecules, and you can use these molecules to further amplify and to get a very robust signal. So these technologies have now scaled up and given place in many ways to very different technologies for really scaling things up, either through cell sorting, where you can basically sort individual cells and then put them in wells, or from microfluidics, where you can guide individual cells into the right place for profiling them, or even pipetting individual cells and then profiling the RNA of those individual cells by amplification. And this is what you get when you see individual single cells on the rows here at different points in time, and different, individual genes across cells at different columns here. So you can see across the time course of different responses, how the individual genes are turning on or off. And you can see some places where the genes are on for all of the cells, others where there's actually heterogeneity even within particular time points. So if you start now looking at individual cells on the rows and individual genes on the columns, you can basically see that if you look at 10,000 cells together, you see what the RNA signal looks like. And you can see here the difference between exons and introns. You can see that most of the signal is coming from exons. This means that we're capturing processed RNAs, but in some cases, we will also capture the unprocessed RNA where the intron is still there. And you can see that for many genes, you see this very nice agreement where individual cells basically have sufficient numbers of RNA molecules that the shape of individual cells, in fact, recapitulates the shape that you see across a group of 10,000 cells. But for other genes, you have a very different picture where, yes, some cells indeed show robust expression, but other cells show zero expression for those genes. And the reason for that is that those cells might simply not be expressing that particular RNA molecule at that time. So there's a lot of talk about the zero values of single cell data. And some of that is indeed technical because of, bursting, or because of not being able to capture all of the RNAs in a tiny little prep. But many of those are actually biological because of the variability between individual cells. And a lot of the challenge with single cell analysis is distinguishing those two. But what's really interesting is that if you take genes that are robustly expressed using single cell technologies, and genes that are partially expressed and genes that are rarely expressed, and then you make probes using these fluorescent in situ hybridization technology, where you hybridize a probe complementary to the gene of interest, you can basically see that indeed there's intermediate expression here, strong expression there, and strong but rare expression there. So in fact, what we are seeing in our single cell data is in fact recapitulating what you are seeing in biology. And these are the corresponding distributions of abundances at the single cell level. So when you stimulate those cells, they respond differently. And what you see is that there's dramatic differences between the housekeeping genes, which show very little variability, and the most variable genes, which are very often involved with environment sensing, defense response, inflammatory response, and so on and so forth. So this is the traditional sort of original technologies for probing individual cells. And these are basically sitting here. These were the first technologies for, profiling individual cells at a time. And then since then, we've seen a dramatic transformation where we have scaled by several orders of magnitude the number of cells that can be studied in a single experiment. And the three technologies that I'm going to be focusing on are going to be SmartSeq, which is using cell-based technology, DropSeq and 10x, which are using droplets instead of, this is well-based. This is using droplets instead of wells. And then SplitSeq, which is never actually separating individual cells, but figuring out other tricks for barcoding them. So the traditional approach had been, okay, let's put individual cells in individual wells using a pipette, or through microscopy, then through fluorescence activation, cell sorting, which is basically shining a laser as cells are flowing through. And then when cells that have particular markers are detected, then you select them for single cell profiling. But you can also do that without selecting for particular markers, just as a means of separating individual cells from each other. You can also use laser capture microdissection to basically go and capture with a microscope individual cells and then profile them. You can use microfluidics, which is bringing the cells from one side and then droplets from the other side, which are forming in an oil medium, enabling you to now trap the cells together along with barcodes in beads that allow you to capture the identity of what cells of what cell every RNA came from. So all of the RNAs from the same cell will be labeled with the same barcode. And then there's, of course, blood collection directly where you can capture individual cells. And the basic technology is, the basic pipeline is the following. Basically what you want to do first is separate the cells. So basically trap the cells inside hydrogels or other ways of doing that. And that's what you see in DropSeq and a lot of these related methodologies. And then you want to amplify the RNA, and you want to sequence, and you want to use different types of analysis after that. And as your fact sorter, for example, might be selecting different populations, you might be inserting them into microfluidic devices or capturing them directly into droplets. So let's talk about these individual technologies and the advantages of each, focusing in specifically on SmartSeq, DropSeq, and then these pooled approaches. So what does SmartSeq do? SmartSeq basically uses fluorescence activation cell sorting. It captures between 50 and 500 cells based on how many wells you're willing to profile. And then it costs about $3 per well. And you can profile, up to 10,000 genes per cell. So this captures the most genes for each one of those cells. But effectively, you're paying a separate sequencing reaction experiment for every single well. So effectively, you're paying for 500 experiments to profile 500 cells. So who's with me on this technology here? So let's see. So the concept is you're separating every single cell into a separate well, and then you can do well-based experiments the same way that you've always done them by amplifying the RNA, and then you have unique IDs and addresses for each of those, and so on and so forth. All right, so 59, 36, 500. The second technology is DropSeq or droplet-based technologies. So here, the idea is the following. You're going to be flowing your individual cells through the one microfluidic chamber, and then you're going to be attaching them with barcodes for each of those, which are attached in beads. So every bead has hundreds of thousands of copies of the same barcode. And within this microfluidic device, you're effectively capturing individual cells along with their beads in a single droplet. So effectively, the droplets become your wells for your sequencing reaction. So you're now capturing individual cells. Instead of capturing them in wells, you capture them in droplets. And every droplet has its own bead with a ton of barcodes. Who's with me on this part here? How we're replacing the wells with droplets. And those droplets are basically serving as a way to separate individual cells from each other so that we can label all of the RNAs of the same well in the same cell type, or sorry, in the same cell identity. So we're at 80%, 20%, 400. So the next step is very cool. You now want to avoid doing a single sequencing reaction for every well. So what you're going to do instead is very smart. You're simply going to now capture all of those individual RNA molecules that have been barcoded with the corresponding cell identity in a single big tube, everyone labeled by the cell that they came from. And those cells that have been labeled with the red barcode will have a red barcode on every one of their RNAs. But then you can separate the, you can sort of break the partitions and just lump together all of the barcoded RNAs and just do a single sequencing reaction for all 10,000 of your cells. And the main difference here is you're doing a traditional RNA-seq experiment, but you have managed to label which cell every RNA came from. So you basically have red barcodes. You're like, oh, they're all coming from the same cell. And you have green barcodes. Oh, they're all coming from the same cell. And then blue barcodes. Oh, they're all coming from the same cell. Okay. So after the fact, after doing a single RNA sequencing reaction, you can then go back and assign individual cell identities to every one of those RNA molecules. Okay. Who's with me on this one? That's an important concept. Awesome. So 64, 27, 550. So that's the second technology. And that's a very, very cool technology. And that's, I would say by far the most widely used technology. So basically, 10x Genomics, for example, has embraced this, and they sort of manufacture those kits that you can use to, carry out hundreds of experiments in individual labs. All right, the third type of technology, and now you guys are going to love this one, is split-seq. And split-seq basically says, well, I don't want to do wells. I don't even want to do droplets. I'm just going to shuffle the cells among a bunch of different wells and reshuffle them at every iteration. In the first iteration, I'm going to have 100 wells and 100 barcodes. So I will only be able to separate every, 100 different categories. And then I will add 100 different barcodes to every cell. What's really cool here is that every cell has all of its RNAs together as it travels through the first round of barcoding. You then pull all the cells together and you redistribute. You then have, for example, the blue cell shuffle, and now it gets the green barcode. And then you shuffle again, and that cell that now has the blue barcode and the green barcode gets shuffled again, and then you also add the red barcode. So you end up with every cell having a unique combination of 100 times 100 times 100 different choices for the barcode. So in the first round, it gets one of 100 barcodes. In the second round, it gets one of 100 different barcodes. And in the third round, it gets one of 100 different barcodes. So you can then have one cell that is labeled blue, red, red. Another cell is labeled yellow, green, blue. Another cell is labeled green, green, blue, and so on and so forth. So what you end up with is a unique combination of barcodes linearly added one after the other for all of the RNAs of the same cell. I never separated the cells individually. All I made sure I do is that I pull and redistribute all of the cells at every one of those wells. Okay, so let's see who's with me so far. So what I'm doing here is that I'm not creating these droplets. Instead, I'm just making sure that every cell stays together as I separate it into different wells each time. And the particular order of wells that every cell went through allows me to uniquely distinguish those cells from each other. So 73, 23, 500. Why is this very cool? This is very cool because I only need to do one giant sequencing reaction at the end. And every RNA molecule will have a set of 3 barcodes, giving me 1 in 100, 10,000, 1 million. So I can basically have 1 million unique addresses for every RNA molecule. That basically tells me which combination of wells did it go through, and therefore which individual cell did it come from. So again, this is drop-seq, this is split-seq, where you're basically, shuffling all your molecules. And then in the end, every single RNA molecule has a set of 3 barcodes that have been added at the end, enabling you to distinguish exactly which cell it came from. And you can just add more splits. You can add, 4 splits to basically capture 100 million cells, and so on and so forth. So again, there's 3 main technologies. There's the cells in wells, then there's the droplets, and then the combinatorial indexing that we can use for all of these. So now we've talked about single cell RNA sequencing, but you can do many, many different types of assays. You can basically look at single cell DNA methylation profiling, single cell genome sequencing, single cell DNA accessibility, and in particular single cell ATAC-seq, which is the assay for transposase accessible chromatin, which is one of the most commonly used assays beyond single cell RNA-seq, single cell ChIP-seq. And again, we talked about mRNA already with 10x, SplitSeq, and SmartSeq. And then you can do pseudotime analysis. You can infer spatial positioning. And we're going to talk about spatial transcriptomics in our imaging lectures. You can look at different cell surface proteins, different intracellular proteins, and the list goes on and on. So there's been many, many different assays. I encourage you to, read up on those. And the most common of them is single cell ATAC-seq. And what this is basically telling you is for every individual cell, what was the accessibility of that cell? Now, single cell ATAC-seq is very different from single cell RNA-seq. And the reason for that difference is that I have many, many different copies of an RNA molecule inside my cell, but I only have 2 copies of the genome. So that basically means that I can either have the region being accessible in one chromosomal copy or accessible in another chromosomal copy, or not accessible in either of them, or perhaps not detected as accessible in either of them. And that basically means that the data from individual cells can be very, very sparse. But when you lump cells together, you end up with a signal that in aggregate looks just like bulk ATAC-seq or just like bulk DNase hypersensitivity mapping. And that's very exciting. But at the level of individual cell, of each individual cell, you might not have enough information for every one location. But by aggregating across many locations of the same cell, you now have enough information to talk about transcription factors that might be commonly active in one cell versus another cell versus a third cell. So you can aggregate data at the level of transcription factor variability. And there's many ways of integrating single cell ATAC and single cell RNA. And some of your projects are in fact already tackling that. So, and more recently, there actually are methods for single cell multi-omics, which allows you to, for example, separate each individual cell, and then each individual well can have multiple different types of reactions. And you can do that with many of the technologies that we talked about. And there's been, many approaches for single cell multi-omics that are increasingly emerging. So there's many computational challenges in dealing with noise and with doublets, and I've prepared a lot of slides for you guys, but I encourage you to watch one of our previous lectures on this if you want to sort of dive through the different methodologies. But what I would like to do this year with the opportunity to invite people from across the world is actually have 2 different guest lectures across 10 different time zones. So the first is hailing in from Europe, Fabian Thijs, and then the second one is actually hailing in from the West Coast. And if life gives you lemons, you make lemonade. So we're kind of stuck far from each other, but even though the people who are near end up far, we can actually bring the people who are normally far near. So Fabian, it's a true pleasure to have you here with us. Take it away. You can share your screen and start.
Hey, Manolis. Hey, everyone. Thanks for the kind invitation and the fun format. That sounds like a really nifty idea. And yeah, I'm looking forward to sort of outlining in 20 minutes, which I'm starting now, what we have been contributing, but maybe sort of what the field is a little bit doing deep representation learning and single-cell genomics. I guess the lecture is, what's it called? It's called Deep Learning in Biology. Is that about it, roughly?
That's the title of the course, and we're going to be covering so many different topics.
Yeah, I saw a bunch of the videos, and that was actually pretty cool. I like it a lot. This is a master class, though, right? No bachelor, I guess. Because I was impressed. I did study at MIT ages ago. I did take class. I was actually at Brandeis University. Loved the place. Really loved being in Boston, but unfortunately not to now. So you have to just look at me. I will briefly go via introduction. I don't know if you showed that, Manolis, but this is the idea. We have the smoothie in bulk, and now we sort of pick apart the food salad. Might have seen this a couple of times. Reason being that, for example, droplets, but also microfluidics have been sort of really being combined with very fast tagging, so that this has become rather readily, broadly available technique. I won't go in those details because you heard that already from Manolis, but I just want to say that this has been quite a big thing. Nature Methods called Method of the Year already a bunch of years ago, and it has been followed up with all kinds of additional essays. You guys can hear me, right? Things working out?
It's a little low, but we can hear you.
Yeah, unfortunately, I kind of lost my
it's okay.
iPad, but it should be fine.
As long as every student cranks up the volume, I think we can hear you extremely well.
Yeah, I'm trying to speak up. So this has been called Method of the Year with the multimodal version just 2 years ago, and now the spatially resolved one. So always single cell. So no wonder this has become really huge. And this was sort of my main original intention, that I've been sort of always interested in machine learning, but in those days when we wanted to do something in regulatory genomics, stuff was always called small n large p. So the statistical aspects were just always regularization and a bunch of linear models. Now we are in this regime where we have 100,000 of samples. That stuff is like super sparse and all kinds of noisy issues and so on. But still, the covariance matrix has become full rank. Suddenly there's this whole world of new avenues that become possible. So it's a big data discipline if you want, and a lot of fun can be had. So how do you do single-cell transcriptome analysis? And there's a whole bunch of pre-processing things, and it's quite exciting and interesting that we had to redo simple stuff such as clustering it again and again, even though you should have thought that this had been solved 20 or 50 years ago. But there's big peculiarities that sort of need to be adapted. So we've been reviewing this a bunch of years ago, where essentially, you first do data processing. Now that they're so commonly available, you have a bunch of count matrices, genes times cell match, and you see now they are longer, right? So they used to be tall. And then you have to do all kind of pre-processing, such as QC, potentially look at data correction, potentially across batches, often very nonlinear things, normalization, feature selection. And then typical first step is that you visualize whatever you have as an outcome, right? So you make a two-dimensional, often nonlinear dimension reduction. Initially, we had a lot of discussions what would be best way to do that. But then you do some clustering on top of that. So that's sort of the first unsupervised big thing. Remember, in contrast to, let's say, variation that we see in population cohorts, we don't have a particular single information about a cell, but often only just about cell ensembles. So this is an inherently unsupervised learning problem or representation learning problem, right? So simplest thing, just find clusters in the data. And a lot of the early papers have been dealing with what would be an ideal method to do so, sort of comparisons and things like that. In any case, once you have them, you want to identify them. And then this whole annotation part is now a more supervised thing. Let's say you have an old annotated data set, you want to transfer that type of things. So quite interesting questions here. And then down the road, you do trajectory inference, composition data analysis, just plain old differential expression. And even these type of things, they're all sort of somewhat special because, differential expression used to be that we just do a t-test or maybe something a bit more fancy where t is secret, you sort of adapt it to a negative binomial. But in this case, you have to do a bit more because you might want to test for differential, for example, changes in variance. This is, I think, an interesting aspect, and my lab particularly has been investing quite a bit in looking at time series information in that data. You just have a static picture, but if you align things, for example, by similarity, you could learn something about cellular differentiation processes. And that has been already one of those early times where people have been doing representation learning in a bit more complex fashion adapted. My lab has been contributing a bunch of things that one of the sort of more tool or frameworks what we've been doing is called ScanPy, single cell analysis in Python, which essentially gives you sort of a library of tools and adapted modules where you can do many of these steps, and has become tremendously successful. Really happy about this big user base of that. But I should say, right, visualization, then this part, but then also downstream, this is all essentially latent space learning. And most of the current techniques factor across the latent space part. So you want to have a method that represents those things well. let me say that the de facto standard, what's mostly done is a KNN graph. So you just look at similarities of cells, and then you do, for example, community clustering. It's something that's very popular. What I want to talk today about, because that's a deep learning lecture, right, is that with this increase of data sets, or this explosion, people really have been starting to look also in neural network techniques for that. And for example, Nir Yosef, my lab, as well as many others. So we've been sort of the first to push this in that field, and there's been sort of many follow-ups where essentially you take an unsupervised architecture. And I think you all have seen this type of autoencoder architecture. You feed in data, such as gene times cell matrices here. Then you squeeze it down, you blow it up again, so you make them go through this bottleneck, what it's called, right? Then you have a loss function, you make the output as similar as possible to the input. Then if you do that, and let's say everything was linear, what would you get? Class? Say this was an architecture, we have just, everything is linear, just linear weights, and we have a mean squared error loss. What would you get in that layer? Principal component analysis, right? Remember, what's the optimal encoding in a lower dimensional fashion with respect to mean squared error? Well, that's the first bunch of principal components. So it's kind of cool that, you know, this autoencoder generalizes the PCA type of framework. It generalizes because, well, it adds nonlinearities here, and then in particular also you can change the loss functions here. So that's sort of a very nifty picture of nonlinear PCA, if you want, and a parametric version, which is nice in comparison to, let's say, UMAP or T-SNEs. So in any case, what you can do with this thing, because it went through that bottleneck, you can actually reconstruct stuff. You can, for example, impute information. I'll say something about that in a minute. But what's very interesting is that you can also look at this bottleneck layer. As in the PCA, there would be PCA space, and some of these things might make sense. For example, if you have a differentiation process going on here, where we just see a snapshot of these cells doing something asynchronous, such as cell differentiation, well, you might actually you might pick up this type of differentiation sequence in here. So in any case, many of those variants around, they won't go through all those details, but what my lab, for example, has been doing, we took this plain autoencoder architecture and tried to apply it for denoising. As I mentioned, right, this stuff is really noisy, so we adapted the cost function. Mean squared error doesn't usually cut it for these things. This is count data, right? So for count data, it doesn't make sense, maybe perfect sense, at least unless it's really high values, to look at mean squared error means essentially output distribution being a normal distribution, right? So maybe you replace this by Poisson, or in this case, because we know it's sort of a bit longer tailed negative binomial, or maybe even a serial inflated one. So what we generated, this was work by Lucas Simon, who moved on to be professor in Austin, and Göttchen, who was actually in Boston at the Broad, but now moved on to Genentech, as a bunch of quite famous people I heard did just recently. But we developed this thing called deep count autoencoder, which is essentially just an autoencoder with an adapted noise function here, where we replace the mean squared error by a zero inflated negative binomial. And then you just model mean dispersion and dropout. In practice, actually, very often we leave out the dropout because just for droplet type of data, negative binomial is fine. And then you get that. And for just now, this toy data, I won't show you many examples. In this case, we have ground truth, just a bunch of now just two-dimensionally visualized cell clusters. Then we add some dropouts, we make it noisy. And if we denoise this type of thing with a deep count autoencoder, we get the cluster structure more or less. Obviously, all this is not unique up to rotation and so on, but you get the clusters. Whereas if you just use the mean squared error, you can't pick up those things. So it does make sense to adapt your whatever machine learning model you choose to the particular data set. So that was fine. But and people have been using it for that. It's not super trivial when you should actually denoise and when not. But what I was most interested down the road in this thing is that this bottleneck layer was actually doing interesting. Oh yeah, I should say that this method, because it's a neural network, right? It scales like hell. And that's like really, really nice feature. Those things that are based on KNN typically have a scaling behavior that is not super nice because, they need to sort of go through pairwise differences, whereas, mini batch gradient descent type of method just really scales well. So that's a nice property that you just get out of the box. But what I wanted to say is that this bottleneck layer, that turned out to be really interesting. So what we've been, for example, doing, we've been looking at a PBMC data set. These colors would be just different cell types. And we've been really squeezing it through a two-dimensional bottleneck layer. In practice, you usually keep this much higher dimensional. So you start with 20,000 input genes and you just go to, let's say, 50 or 100. But in this case, we just went to do 2. And then you get such a plot that pretty much looks like a T-SNE. So it finds those cell type groups, which we didn't tell it about. So this means that this bottleneck layer seems to learn something about this biological process about, in this case, the cell type clusters in blood, right? So we've been thinking, can we leverage this information in the bottleneck layer? And that's the idea. So, what we often do, I don't know how you do this, Manolis, but we often just see what the computer vision community has been doing, because they've been sort of playing around with so many things already. Maybe there's an idea to pick up.
I have to admit that our first 6 lectures used machine vision as a very common example.
Yeah, it's so popular and it's so visual. And I should have said this as well. What's cool in vision is, of course, that you can do convolution. So you can sort of reduce the parameter space of all the parameters of neural network to sort of stuff that's sort of translation invariant and scalar and so on, right? Which in gene regulation we can't do so easily. So often our networks are more connected and not as.
But again, it's up to you guys to develop the next generation of convolutional filters that actually apply to genomics and single cell data, etc.
Yeah, if someone wants to do that, I have one project actually going on with a colleague in Tübingen in this direction. I think it's super hot. And if you have ideas about that, Manolis, I had all the times these network smoothing things. I've been trying that out. We never had excellent performance. We didn't push it so much, but I think it could be now with this huge data set a good time to do again. So
we pair students up with some of the guest lecturers. So if you're willing to supervise some projects, I'm sure students will be thrilled to work with you.
In these virtual days, it doesn't really matter. So yeah, I'd be very interested and happy to talk. So let me just get this idea across. I thought it's a very nice one. You might have heard about domain adaptation. So just sort of taking a setting and transferring the style to a new one. For example, painting style. In this case, it's a data set in computer vision trained on faces. This guy representing now the class of men with glasses. And what they did in this thing was arithmetics in the latent space. They subtract a man without glasses and added a woman without glasses. And lo and behold, you get samples from women with glasses. That's pretty cool, right? So you transfer this meta concept where if you were doing this on the pixel space, because of the nose, eye, whatever ratios are different, this wouldn't work at all. It works in the latent space. So we've been asking ourselves, what would be the complement of that for cells? Let's say we have a cell that underwent some perturbation, a drug stimulus. Let's say an overall disease or whatever. We subtract then that ground cell type in the ground state and add a different cell type. And could we predict then this cell type? We could predict, or we could interpolate. We can try to interpret. So that's sort of a prediction type of problem where we now leverage perturbation as with glasses. And we've been thinking about that a bit. We could also sort of say maybe more complex style transfers even across a species and so on. And that would be the architecture, just latent space arithmetics. So the idea of this thing we call SCGen was worked by Mo together with senior postdoc Alex, who moved on to a startup that's actually also Boston-based, Celeri. I should mention I'm consulting sometimes for them, so I don't want to make advertisement for them. But in any case, this SCGen is now trying to model these perturbation effects. So let's say you have unperturbed cells and perturbed cells here, and you have a bunch of those. The question is, how would a new cell type behave? And you can imagine if you were able to do this robustly, you can sort of amplify and quicken all kinds of drug screens where, if you want to have space-specific response, this is something that doesn't scale up to do manually. So if you can do this in silico, that could have potentially quite some power. So what we just do is we encode it. We don't use the plain DCA type of architecture that I showed you before, but a generative version. Then we go into
Fabian, let me ask a very interesting question by Steve, who's basically asking, do you do this for every cell type, for all of the cells jointly? At what level do you apply this representation learning?
Yeah, it's very important. So what we feed in is really a matrix where each cell gets a separate column. So, essentially in the gradient update to learn the parameters of a neural network, each cell is separately fed through. There's no way we can really group cells except for cell types, but then we sort of bias the data with respect to some type of pre-clustering. And there's some people actually doing this. There's some reason also for that, but we don't group them. So it's really per cell, but it's good to have variation in there. So if everything was just a normal distribution where things have been sampled, it's going to be boring. It won't find a good representation. So it needs to see a lot of variation, such as cell type, but there's also, you can't imagine how long the community, I'm involved in this human cell labs, have been discussing about what is a cell type. Because it turns out these things are sort of fluid, right? And sometimes there's a cell state between just cell cycle stage and so on. But in any case, if you have variation, you just try to leverage. Yep. So what we try to do here is really also, we feed in all of these data sets, encode them, and then we hope that we get such a linearized latent space where we can do these arithmetics as you saw with the images before. So we learn then the perturbation effect. And in this case, it's really literally, because we essentially apply this linear arithmetics that you saw before, that you assume that you get one direction that is dominated by the perturbation effect. And then we apply this to the new one and we just decode. And that's astonishingly powerful. So this was really working out, right? So this is what we call SCGen. It's a variational autoencoder with those latent space vector arithmetics. We can actually do out of sample prediction with that. We've been just recently extending this in a collaboration with Facebook AI towards a model where we can actually really do more complex, really latent-based decomposition. But for now, we're doing that. And just to show you that this actually works, let me show an example where we have PBMC data sets unstimulated as well as stimulated. Okay, so this is the control, this is stimulation. Colors will be indicating cell types. And then we ask, can we reconstruct a cell type, such as in this case, the CD4 positive T cells, in the stimulated fashion by just leaving them out? So we really do what's called in machine learning out of sample prediction, right? And that's not potentially trivial problem. And what actually turns out that the prediction really fits not only in terms of mean, but also roughly or not so bad, at least also in terms of the distribution of the variance. And we can do this across all the different, in this case, each of the dots will be a gene, and we see we do a very good job across all kinds of stimuli. So if you compare our prediction with the real one, we can reconstruct this one very well. And then we do this systematically, not only for the CD4 positive T cells, where you could argue maybe it's just a linear shift, but we do it for all of the different holdouts, and we see that we can reconstruct the different cell types robustly. And then what's interesting is that if you now zoom in and check, actually, for example, in this case, we look at the reconstruction in the CD4 positive T cells that I showed you before, then this ISG15 was reconstructed. But for example, this ApoBec3A is just not even expressed in this positive CD positive T cells, so you also don't actually estimate it. Whereas in the dendritic cells, this sum is sort of okay, but you also reconstruct this ApoBec3A, which is different. So and this is really a key point. We have not learned then a linear signature, because this signature is different from this one. You don't learn a linear one, but you learn a cell type specific one. It's just linear in latent space, but because of the decoding, it gets non-linear in the gene expression space. That's why you can have cell type specific responses. And I thought that's actually quite nifty and cool property. I think there's potential extension, because you just saw this was just for RNA, so you can now combine this with ATAC and other type of double measurements. You can combine it with spatial, where you can actually then add convolutions and so on. So I don't want to take too long, so let me just show you two more slides. So there we had a bunch of applications in the paper, and there's some limitations. So we've been currently working on extending towards multiple perturbations, also going beyond linearity in latent space. But one thing I just want to say is, because this field has become so popular, that a lot of people have been generating something like, this is called human cell atlas, so like a periodic system of elements, but now of cell types. Because in the end, when we do genomics, we don't recluster, we don't re-annotate each new DNA sequence anew, right? We just transfer that information. So de facto, if you want, and it's sort of a bit far-fetched, but in genomics, we kind of do transfer learning, right? So once we have a gene annotated, we just reuse that information. We'd like to do the same thing for cell types. It's not yet happening, but those atlases are being generated. And I'm, for example, involved in the lung atlas, and we've been applying this also to COVID and so on. So there's a bunch of nice papers in collaboration actually with a bunch of people at the Broad. But there's some challenges in using those atlases, because each of those maps look different. How can you also deal with distributed data? All of this is human data, so you might not be able to share this robustly. And how can you make those maps easily available? You want to have like a Google Maps, but without sharing each of the individual sampled cell. That's going to be too much. And for this, we've been coming up with the same neural network type of idea. And that's what I want to show you in this thing. So it's an extension. It's called SEGen. It's just out there. And I won't show you examples for that. I'll just show you the concept. We take reference data set to the same type of learning that I showed you before, except for that we now add a condition that labels each of the studies. It's called a conditional variational autoencoder. Let me just upload this thing here in a repository. So your cell type map is just the encoder, or maybe the decoder, depending on what you want, but it's not the samples. So all of the information of the big sample is just packed into that model. And that's like super useful, because then you just can throw away all the samples. And once you have this, you can do your style transfer. And then in this case, because the studies are packed in there, we need to do what we call architectural surgery. So we add query labels. Won't go into details with that, but essentially we can share those variations as well, and we can actually apply this. So this is our idea of transfer learning in this setting. I'll skip that. We have a nice model zoo for doing this as well, but I preferred really talking to you a bit more slowly. So let me just sum up. I think latent space learning and single cell genomics is really a fun area, because you can essentially use all kinds of extensions of autoencoders, but of course also of really generative models. SEGen was one of those simple generative models we've been doing at the time, and we've been extending it with latent space learning. And if you're interested in trying out things, but you don't want to sort of really get into single cell things, we've been coming up with a model zoo. And I heard you had Anshul presenting just a week ago, right?
Yes, indeed.
And I don't know if he mentioned his Kipoi thing.
No, he hasn't. But I'm actually classmates, believe it or not, with Julien Gagneur. When we were
really
13 years old, we were classmates in the same class.
No way. Small one. He has mentioned you at the time, but I didn't know it was from classmates. Yeah, Julian, that's a big thing for him. So we've been talking with him a lot. We've been trying to do something related for single cell expression profiles. And this Spira, which is Greek for sphere, so we wanted to mimic the Kipi thing, right? Gives you sort of very readily a bunch of data sets to try out whatever fancy ideas you cook up. And we have some extensions towards spatial data and interpretability. Won't mention that. This is how Bavaria looks all the time. And, we had to do social distancing. But yeah, at some point, it'd be lovely to actually see each other physically. But that's about it. Thanks.
Fabian, thank you so much. This was really phenomenal. And I know that we're low on space, but since you mentioned Kipoi, which means gardens, and Sphera, which means sphere, I have to say that it is today the 200 years of the Greek Revolution to the day. So,
I heard it in the radio this morning. Yeah, true. Oh, cool.
And Romain might actually be on the West Coast, but he's actually French. And I just have to say that, European unity in the face of adversity is a great thing to celebrate today between the Greek, the French, and the German. Anyway, thank you so much, Fabian.
It was great seeing you guys. I go back to family, so I need to log up.
Good luck with that. Bye-bye. Salut, Romain. Welcome.
Hi.
So take it away. Just share your screen and jump right in.
All right. All right. Yeah, thank you, Manolis, for inviting me. I've been looking a bit over, like the website of the class, and I wanted to say that I would have been really excited, 5 years ago to take a class like this.
I have to say, I would have too, and that's why we were making it.
So this is great. And today I will be presenting some of my work, which is very related to what Fabian's doing. I guess maybe this presentation will be getting a little bit into more details of the Bayesian modeling and applied statistics, which is what the side that I've been taking on for my PhD. But I'll be talking about this application of VAEs to single cell data. So overall, this has been a very collaborative collection of projects shared with graduate students, with master's students from France, undergrads from Berkeley, and professors. So I've been working with Pierre, Adam, Chen-Lin, and Galen, and also Jeff, Mike, and Nir. So Mike and Nir are both of my PhD advisors, and Jeff basically played the role of an advisor and is now a professor at University of Michigan. Today I would like to talk about plenty of things. I think the point is more to slowly go through things one by one, and I hope that you can review these slides if you don't have time to do everything. But I'd basically like to set up the scene for why it's meaningful to apply VAEs to single cell data from the modeling perspective. Then I'll talk about SCVI and plenty of other extensions or failure modes of VAEs that I saw and we try to correct. So let's go. So the key idea of doing single cell transcriptomics is that cells share the same DNA, but they have distinct functions. So if we look at, for example, the H&E stain that we have on the left, we see plenty of different color densities, which mean different type of chemicals, glycogen, etc. in the tissue. But essentially, it means that all these cells are kind of working together, but they are pretty heterogeneous. And so we can have a lot of questions, right? So we want to understand what type of cells do we have in this tissue? What functions does this cell carry? And essentially here, it's a multiple sclerosis model. And so we'd like to know how does it change from the healthy to the inflamed tissue? So single cell helps having some type of answers to this question. The way it works is you can dissociate your tissue into single cell, for example, the inflamed tissue and the healthy tissue, and then you can run single cell RNA sequencing pipelines. There are many of them, but you get a result that's on the right, which is basically a matrix of counts that gives you for every cell how many times you have captured the transcript aligning to a given gene. And so what makes it really exciting for me, especially when I started grad school, the first thing as a computer scientist, what was exciting was that the sequencing of 1 million cells from 10x Genomics that showed that we needed to make algorithms that scale. And the second thing as a statistician that came out and was interesting is that we can sequence plenty of things, or we can measure plenty of things from the single cells, perturbations or protein and transcriptome and plenty of other things. And so, yeah. So there are plenty of the biological questions are formulated before we can cast them into algorithmic queries, which are like not exactly defined, but we can go from this into the mathematics and Bayesian statistics. So basically, we have cell level queries and gene cell level queries. The way that the cell level queries work is we'd like to, for example, stratify the cells, right? We don't know what the cell types are. Or we don't know how to identify the cell state. So we'd like to project the cells into a low dimensional manifold. And that's right. And that's when you hear PCA, VAEs and all these type of things. But we'd like to do maybe more refined analysis. Some people do trajectory analysis and plenty of other things you might have seen in class. Another important task is harmonization or integration of several data sets. So I'm sure that Fabian talked about this type of problems. Essentially, we have data set specific biases that makes like measurements from single cells maybe not directly relatable from one data set to another. These are called batch effects. And so we'd like to provide an embedding that is relatable across different experiments. And then we have the annotation problem, in which we'd like to, for example, one data set is annotated, and it's pretty hard to do it. Pretty hard to do it in a good way. And so once we finally annotated the data set, we'd like to transfer the annotation to other data sets. And then with respect to gene cell level tasks, one of them can be referred to as normalization or imputation. It's basically the idea that I showed you these matrices of counts, and most of them are zeros. They are sampled from a stochastic process, right, a Poisson or a negative binomial distribution. And we'd like to know, for example, the mean of that distribution to get a better idea of like what is the underlying distribution. And more especially, we'd like to get the snapshot of this distribution, but and how it modulates with technical effects. So for example, removing the technical artifacts. And if we're able to do this, then we can do the most important thing somehow of the gene cell level queries, which is differential expression. So for example, we have two cells, or two cell types, I guess, two bags of cells, and we'd like to know which gene is significantly different from one another. So that's a hypothesis testing problem. And I'll be talking about all of those problems today. But the goal is to do this, with in a probabilistic way. And how we're going to do this, where we're going to build the latent variable models. So single cell analysis can be complex for several reasons. The first one is, we have technical noise in a good amount. So the first technical noise that I'm going to talk about today is variable sequencing depth. So maybe you've been seeing this in class. It's basically the idea that from a given cell, you can get 1,000 transcripts or 10,000 transcripts and sometimes even 15,000 transcripts. And it can give some biological information, but most of the time we want to correct for it because it can be a confounder in differential expression, for example. The second type of technical noise is batch effects that I already mentioned, this data set specific bias. Then what's an important point to raise is that the data comes from this multivariate count distribution, which is high dimensional. And we're in a regime that the mean of the distribution is so shifted towards zero that makes the measurements really non-Gaussians. So we want to think seriously about what type of distributions we put, in these generative models. And the last thing is, as I said already, we need scalable methods, and we want to keep in mind that the scale is millions of samples. So there are many standard machine learning methods that are applied to single cells. Some of them are also more traditional statistics methods. It all overlays, but I'm going to present a simple, like a stereotypical or archetypal, I guess, pipeline. So first, we have the counts. We want to normalize them, and there are many ways to to do these type of things. But let's say we just divide by the total number of transcripts in a given cell. We apply some logarithm transformation to make the data look Gaussian somehow. Then we can apply a PCA to reduce the dimension of the data. We can apply an algorithm to correct for batch effects. So many are, for example, like creating a graph in one data set with the PCA, another graph on the other for the other data set, and do some graph matching. These algorithms are really popular in the field. Then we can cluster, like the corrected latent space to identify cell states. And finally, we can go back to the raw data, the counts, and perform differential expression to give names to the cell states, CD4 T cells or like subset of T cells, inflammation, etc. And somehow, so what struck me from like 5 years ago when I learned about single cell was, why do we need to like change the data so much? Why do we need to apply different algorithms that maybe have different assumptions? For example, the differential expression builds a negative binomial model, which is really nice. And why can't we recycle it to do all the other things? So the key idea is, can we find a unifying modeling assumption for the whole pipeline? Which was very attractive to me as an idea of something to work on. And where things start is looking at the PCA. So what I write in equation 1 here is a generative model for probabilistic PCA. And you can already see how you can improve this type of models. So the first thing I write is so marginally P of X is a Gaussian distribution when you integrate Z. And we know that the expression levels are not quite Gaussian. So if we would like to fit this model, we need to normalize the data, etc., which we would like to avoid somehow. And the way we can do this is replacing it with a count distribution and applying some specific inference scheme. Then the second point is it's hard to assume linearity between the mean of the gene expression levels and the latent variables, especially because, you can have a case where two genes, you have some nonlinearity in defining cell types, right? If this gene is on and that one as well, but not this one. So we'd like to extend this linearity assumption. Then the last thing is that when people apply PCA, it's actually, what you get from PCA is equivalent to solving PPCA with when sigma goes to 0. And we lose the fact that we were working in the first place with a probabilistic model. And this is fine for many applications, but some of them actually, it's really really important to get uncertainty over the measurements, especially for differential expression. And this will come into play later. So the room for improvement is to build a scalable and consistent framework for analyzing single cell data in a probabilistic way. And that's all the idea behind a CVI, to build a deep generative model that you can call a fancy PPCA to basically solve this problem. So now I'm going to go quickly through a couple of slides of Bayesian modeling. And the idea is that this is really what I used, to talk with my collaborators about a CVI, etc. And so I'd like to present it as a legacy for the work. And most of you will already know what a graphical model is. So here in this graphical model, all the nodes are random variables, and the edges encode conditional dependence. So here, basically, the way you can read this graph is you want to decompose the joint as a product of every node condition on its parents. Graphical models are interesting because you can encode many probabilistic properties. For example, if you remove an edge, then you add in conditional independence and you restrict, the class of distributions you're working with. So this is a simpler model than before. What you can do as well is, of course, you can add constants. These constants can be learned along the way, and they can become parameter of the likelihood. You can have independent replication. So in a CVI, for example, all the genes are independent, conditionally independent on the latent space. Then some of the nodes, we don't observe everything, right? Actually, if you observe everything, then I'm not sure you need quite a model. But so here, some variables are latent and some of them are observed. And the idea is that if you know the model, then what you'd like to do is calculate the posterior, right? So what is the distribution for my variables I didn't observe? And you can use Bayes' rule to calculate that. But of course, when you use Bayes' rule, you have a denominator, which is a marginal likelihood. This marginal likelihood is intractable for many cases, and actually most cases, except PPCA I showed you, because it's a Gaussian distribution. And so this is a topic of MCMC methods as well as variational inference. And in SCVI, we use variational inference. That's what I will present today. So the key idea behind variational inference is that for every observation, every observation X, we'd like to calculate the posterior. And so we can't calculate it, we're going to approximate it. And for that, we're going to place a family of distributions BQ, and we're going to find the point Q star that minimizes a certain functional, here it's a KL divergence, to the posterior. And essentially what we're doing here is we're casting the inference problem into an optimization problem. So now, so far we don't know how interesting that is because we don't know how tractable the optimization problem is. So because we choose this KL divergence, it becomes a somehow easy optimization problem. The idea is, so you look at equation 2, we want to find Q star that minimizes the KL, and the Kullback-Leibler divergence actually is the expectation of the log ratio. So, and the logarithm is a morphism, right? So the ratio comes a difference, and you get to equation 3. And then you can use a definition of a conditional density to get from equation 3 to equation 4. And what you can notice in equation 4 is that we have this evidence term, log P of X, that doesn't depend on the variational distribution. So we can take it out of the optimization problem and not change the argmin. And then we get to equation 5. And equation 5, interestingly, everything is tractable because we know the model. The model is fully specified, P of Z and X, the joint. The variational distribution is something we control, and so we can solve this more or less efficiently, but we can, it's something we can solve now, and that's why variational inference got very attractive. So now how does it fit into VAEs? Well, so for example, here I'm going to present an extension of probabilistic PCA in which we remove the two assumptions. So one of them was we had a linear function to define the mean of the Gaussian distribution, and here we can specify it with a neural net. So that's what we have here on this part, and we can specify the variance with another neural network. And the way it looks like, so this is some astronomy data. You take a point in the latent space, and to every point in the latent space, you map a mean and a variance. And then to simulate new data, you can just sample from it, and you get this image of a star. So I wish I had put single cell data here, but it just doesn't look as nice. We don't have those images, right? But yeah, that's basically the intuition for what the VA is doing. And now we're stuck with another problem. We can't just apply VI as I showed you before. The reason is that we need to learn the model parameters as well. The way we do this is we maximize the evidence, so log P of X. And of course, we can't maximize this, so we need to decompose it using the variational distribution. And here from the inequality from left to right, I applied Jensen inequality, but in your class, you just saw a complete decomposition of the log evidence using the variational distribution. These two are very, very similar point of views. And the idea is that what we have on the right here is something we can code in PyTorch or in TensorFlow, and is very straightforward to optimize. So now we have another hic, which is that we have several observations, and for each of these observations, we would need to get a variational distribution, right, with parameters. But what we do in the VAE is that we tie up all the parameters of the variational posterior using two neural networks. So if we have a Gaussian variational family, we need a parameter that ties up the mean and another for the variances, and all of them are just a match from the input data to these parameters. And it's been called many names, and one of them is amortized variational inference. The other is autoencoding variational Bayes, and that's what's used in the VAE. So now we can talk about SCVI. So SCVI is basically taking all of the like technical effects I talked before, incorporating them into a graphical model, and that's actually a VAE. So we want to write a generative process, could also be written with the graphical model as I showed before. The idea is that we have the gene expression counts for a given cell in a given gene. We have the batch identifier, and the generative process is as follows. First, we sample an embedding for the cell, which uses pretty much the same prior than probabilistic PCA or the other VAE I showed before. And you can change this prior. I will talk one example later. Then we have a scaling factor. So this is a scalar number L that is sampled from a fat-tail distribution to account for the many variations in the number of transcripts in a given cell. That's what we call the library size. And we have a neural net that maps Z and the batch identifier to a quantity that I call rho. Rho is a normalized expression, which means there is a softmax nonlinearity at the end of this neural net, which means that rho is a vector for every cell that sums to 1, and it gives you the expected frequency of expression for every gene. And then we can scale this rho with the sample from the log normal distribution, and this gives us the mean of the negative binomial, and we have a dispersion parameter. And that's how we have this, now we have fully specified the generative model. And what I want to draw your attention on, so actually Fabian just talked about this, is that here we use a conditional VAE, which means that we learn a conditional distribution, P of X given Z, P of X given Z and S, I guess. And Z is a latent variable and S is observed, and S is this data set identifier. And that's how we deal with the dataset specific biases. So when we do this, the latent space become invariant to the technical effect, which is a batch effect. So now what's pretty nice with this model is that we have a set of modeling hypotheses, which is common to all the tasks. And so now I'm going to try to convince you that we can solve all the previous tasks. So we were talking about stratification and embedding. Yes, we can do this because for a given cell, we can sample from the posterior distribution or get the mean of Z. I talked about harmonization. So we removed indeed those dataset specific biases because we condition on this batch information. We wanted to normalize, so we did normalize because we condition on L, and we have also this softmax trick at the end of the neural net. When we want to impute, well, we can just sample from rho, and this gives us some imputation. We can take the mean of it as well if we want. And then when we want to do differential expression, well, yeah, we can just formulate a certain Bayesian hypothesis testing problem, and we can based on the rho, and we can solve it. And that's how we will do differential expression in SCVI. So we can do inference over this model as I showed you. And interestingly, it works pretty much like the normal VAE, except here we have a different number of neural nets. We need to have a neural network for the mean and the variance of Z in the variational distribution, a neural net for the mean and the variance of L in the variational distribution, and then one neural net that encodes the normalized mean of the negative binomial. In the original publication of SCVI, we have a 0 and fetid negative binomial distribution. You can just add another neural network. But for sake of simplicity, I just didn't expose it here. So you can look at the paper. So then, if I want to convince that the algorithm is useful, we would like to investigate the latent variables, right? So the first thing is we can look at the embedding Z. And there are plenty of benchmarks that you can look at, so I'll be quick with it. But essentially, even if we have a prior that shrinks toward this normal 0,1, we managed to recover hierarchical clusters. So this is a cell-cell similarity, and all of these are ground truth cell types. We can also recapitulate some developmental gradients. And this is done by just querying the mean of the variational distribution. Then we can look at how SCVI does a good job for batch effects. And actually, so SCVI gets used the most in settings where you have severe batch effects and many batches, and because it becomes an extremely scalable method compared to many competing methods. So SCVI without batch. So here, this is a data set from cells from the retina. Colors here mean a data set, and on the right, colors here mean cell type. So what we would like to do is mix data sets, but still be able to distinguish cell types. So if you look at SCVI, well, it looks good because you can see the cell types, but it looks terrible because all the data sets are separated. If you PCA looks just not that good, but when you look at SCVI applying with the batch, this was with no batch correction, it really mixes the data sets, but we can still see the cell types. And then the other latent variable is RAW. So for this latent variable, to me, the most exciting thing is to look at differential expression. And I worked on this for a while. I'm going to give you a bit of an insight on like why it's a complicated problem. But for the purpose of the first paper, what we did is we had some PBMC data, and we looked at two populations, CD4 and CD8 T cells, and then we calculated I guess this was a second paper. We present you a third one, but we presented the log-fold change, which is a standard metric for calculating differential expression, and we just calculated the posterior probability that the log-fold change between two cells for a given gene in terms of RAW is more than a certain threshold, say 0.5. And this gives you a ranking of the genes, and you can compare how well the ranking, are correct. So correct is always complicated, especially in this setting if you don't have simulations. But for what you did for the purpose of this paper is looking at another technology, a microarray, which is much more stable on the very specific cell types. And we basically compared the ranking of the genes with of SCVI and other methods with respect to the microarray. And we found that SCVI was performing similarly, if not a little bit better, at doing the ranking, which to me was incredibly exciting because that was a proof that we can use the same model to do all of these tasks. And then finally, I talked about scalability. Well, at the time that SCVI came out, so you see here, DCA is a method from Fabian's lab. And that came out really at the same time as SCVI. I guess the only difference is that DCA is an autoencoder and that it didn't use, didn't do batch effect correction at the time. But you see that these are the only methods that can basically scale to millions of cells, of course, with PCA. While all the other methods that are based on MCMC or that invert some matrices or that are like quadratic in scale or blow up in memory, and all of these were stuck in like 50,000, 100,000 cells. So that was a huge improvement to be able to use stochastic variational inference in all these tools from VAEs. So now I'll present. Let me see how much time do I have. Okay. So I'll present an extension that's for annotation. So there are several data harmonization scenarios, and I guess Fabian has been talking about this for a while, so I'll go a bit quickly. But you can harmonize multiple samples from the same lab. That's what I showed you before. You can harmonize data across different laboratories. That's what we see on the right here. And we see here it's a successful harmonization because again, data sets are mixed, but cell types are not. Those are the two objectives. But there is another problem, which is that I didn't talk so far, which is how to transfer labels from one data set to another. And we did this by using a semi-supervised VAE. So there are many methods to do this type of work, but they work with building graphs and matching the graphs and transferring like labels across different graphs, which has a caveat because it cannot perform differential expression. Again, it's not everything in the same pipeline, and we'd really like to push forward this idea. So we built ScanVI, and the way that ScanVI works is that on top of X and S, you can observe or not this C star, which is a cell type identifier. And so NA means unobserved. So the way you can use it for many purposes. For example, one data set is partially annotated and you like to propagate the labels. or one data set is annotated and the other is not. And so the way we did it is we basically extended the CVI by changing the prior in Z from isotropic normal to a mixture of Gaussians. I mean, it's not a mixture of Gaussians, it's a mixture model. And this C here becomes the mixture assignment, like which cell type it came from. And when you don't see which cell type it is, and you have a neural net in a variational distribution for the cell type assignment itself. So you can read this manuscript. There are plenty of extensions of this work, but this was a pretty powerful idea that here I show you one example. So there is a good use case in which you have a subset of T cells in all of the cells, but you don't know what sub-cell types they are. And based on some marker genes that are very lowly expressed, you can maybe annotate a couple of them, but not everything. And then you can use ScanVI to propagate the labels. And it works much better than doing just clustering or just classification, because we can exploit the knowledge about all of these gray cells. It's semi-supervised. So we had the ground truth label from some sorting, and we have the probabilities here that looked pretty reasonable. So then I'd like to give a teaser on another problem that I encountered using a CVI. And this was the first machine learning paper I wrote. So I was interested at the time of a CVI into factoring out things of the latent space, right? So I showed you an example when we can factor out batch effects. And turns out batch effects actually pretty easy in a lot of settings. But what was really hard was factoring out some continuous information or some like alignment errors in other covariates. So I'll present you another example in which, so this is the same problem. How can we obtain a representation independent from a nuisance parameter? The key idea again is conditioning. And so for example, in this machine learning inspired example, computer vision, we have several pictures of different of faces. So person identity is Y, but we have different lining conditions. So all of these angles was a light angle, and we'd like to have an embedding, like Z1, which is very discriminative of the person's identity, but marginally independent from the lining condition. And turns out if you take a VAE out of the box, so it's the same thing with the CVI, right? Like you'd like to find an embedding that is very discriminative of cell types, but independent from cell cycles or other things. And now what you can do is just build this conditional model, but what happens is that the embedding you get are still very correlated with S, with this light. And sometimes it is, sometimes it's not, but for the batch effect it works, but here is really an example where it didn't work. And a CVI with the cell cycles, for example, also suffered from this problem. And turns out it's a problem that comes from the overflexibility of the neural net used in the parameterizing the variational distribution, and it affects all of these issues. So if you want to learn independent representations, if you want to learn invariant, that's what I presented, or even this denoise representation, all of these are very simple variants of what I talked about, and I talk about it in the paper. I have an example for all of them. And what we do is basically trying to restrain the search space for the variational parameters so that we add, in a ad hoc way, we enforce statements of the form, this is independent from that in the aggregated posterior. And the way so it's a bit complicated because we can't enforce these statements just by looking at the lower bound. So what we did in that time is using the Hilbert-Schmidt norm of the cross covariance operator that embeds, like distributions. It's a non-parametric measure of dependence. And we just add it to the lower bound. So it gives you a looser lower bound, but that has more suitable properties. And we call this HC constrained VAEs. So if you're interested in this type of things, I highly encourage you to look at the work. But it was a surprising example where I thought the problem came from the data, but actually it came from the model. So that was interesting. And then I'd like to present talk a bit more about the differential expression. So in the way we do differential expression in a CVI is kind of a Bayesian model selection problem, right? So let's say we have two cells, we have the respective measurements, and we have the normalized gene expression level. For every gene, we can make models, right? Model 1, log-fold change is more than delta. Model 2, log-fold change is less than delta in absolute value. So model 1, this gene is differentially expressed. Model, I guess not 2, model 0, it's not differentially expressed. And what we can do is simply do a base factor, so likelihood ratio. And we can give a threshold. For example, if it's more than 10, this is written in very old Bayesian books. If the odd ratio is more than 10, then you can say that your gene is differentially expressed. And there are a couple of caveats with this. First thing is that the approach is potentially biased, because the way we calculate these posterior probabilities is that we need to plug in the variational distribution in place of the posterior. And so if the posterior is wrong, well, we're in trouble. And the second thing is that the applicability of this type of method is a bit limited, because a lot of people want to see control of quantity they know. So just saying base factors more than 10 is not, it's not as faithful compared to controlling a measure of false discovery rate. And so we spent a lot of time thinking about how to control the false discovery rate doing hypothesis testing with STVI. And somehow those two problems, it's kind of it's a homework if you want, are related to this thing, which is calculating posterior expectations accurately. And so you can see why the first point was related, right? Because we were plugging in the variational distribution in place of this. So, and the FDR is a bit more work. So the idea is we want to calculate this thing, which is, the bread and butter of MCMC methods, but here we'd like to do it with the VAE. And so let's say that we have access to these samples from the variational distribution, and there are a couple of things we can do. So first, we can just use a plugin estimator, right? So averaging everything from this distribution, or we can we can reweigh the samples using self-normalized importance sampling, and weigh those labels by this density ratio, so that we can keep the most promising samples. And these things actually don't quite work out with the vanilla VAE. The first reason is that the model fit by the VAE may not be equal to the data distribution. That's kind of a side thing that we talk about in the paper, but so if you find a better model, it's always better, right, to calculate this differential expression or making decisions with VAEs. And the second thing is that there can be some strong discrepancies between the variational distribution and the posterior. So now it's particularly important because the way that variational inference works is that it underestimates the variance of the posterior. And so if you try to use it for importance sampling, thinking that you get an unbiased estimate of your quantity, well, you have a huge variance because you don't really cover the whole mode, or you don't sample enough, and you're very localized. And this can have disastrous performance for many applications. And so what we try to work on for this work, I guess, is we thought about all the different, like decomposition of the evidence. So this is a decomposition of the evidence you saw in your class, right? Like evidence equal elbow plus reverse scale variational gap, yeah? All these are variational gaps, I guess. And the second, you can also, so this is a lower bound because the KL is positive, but you can have upper bounds. So here we have a certain upper bound called UBO, and the gap between those two things is the forward KL divergence. So this is related to expectation propagation, for those of you who know. And then we have another bound here, which is QBO, in which basically the gap between those two quantities is also negative. So it's an upper bound, and it becomes related to the chi-square divergence. And what we try to think about is those upper bounds are very interesting because instead of underestimating the variance, they overestimate the variance, which is much better for importance sampling. So we came up with the procedure trying to bring those ideas into VAEs and saying, oh, so what we can do for doing hypothesis testing with those VAEs is first we fit multiple VAEs, each with a different variational distribution. We keep the best model based on held out likelihood or any other surrogate. Then for this model, we refit again. We keep the model fixed and we learn different variational distributions. And then we use multiple importance sampling to combine all of those proposals together. And what we see in practice is that, for example, the VAE or IWay is really good at learning the model, but the chi-square divergence is really good for making decisions once the model is learned. And so we combine all these things, and we have some really good results on controlling the fault discovery rate with the CVI and plenty of other things. I don't have much time because it's already past 30. But basically, I really highly encourage you to have a look at the paper if this is interesting to you. And there is a bit of theory in this paper as well, in which we quantify the the error for important sampling using concentration bounds. There is a new VA in there using the chi-VI bound that actually was quite new, and we have some experiments on PC and MNIST as well. So then let's see. Should I stop now? I have 2-3 slides I'd like to go through.
Yeah, but why don't you just cover them and
okay, that works.
Leave if you have to.
Okay. So then there is 2 slides. I think the first thing is we really tried, along all of these papers, we also, how do you say it? All of the algorithms that I showed you today and plenty of others, they were coded in the same code base, which means that we really try to do incremental work and internal developments in the lab to arrive at a good interface with PyTorch and loading single-cell RNA-seq data sets and coding up all these VAEs with the fundamental modules. And we created something called the CVI tools, which for users contains a lot of tools for analyzing single-cell omics data, but for model developers also contains interface to probabilistic programming languages. And so it's very easy to change a CVI in 1 line of code or craft a new VAE. And I think we implemented 10 or 13, between 10 and 13 generative models inside of this code base. So I would highly encourage you to have a look if you think that's interesting. And the last thing is we wrote a review trying to go over all the impact of those VAEs as well as GANs in a modest subset of molecular biology, because it's a really big area. But yes, if you want to learn more, I also encourage you to visit this publication. And yeah, so that's all for today. Thank you very much.
Okay, so thank you so much, Romain. We will, I guess, dive into some of the math and some of these results in recitation tomorrow. So I think folks will have a chance to really ask questions. So thank you so much for really diving into the methodology. And I think it ties in so many different aspects of the course that we've been touching on. And for the students, don't freak out. It's a lot of math, but I think we will cover it in more depth tomorrow. And there will be more opportunities to dive into that. All right. Thanks so much, Romain. Do you want to stick around for just a few moments? And can I have the TA stick around as well? Yeah. All right. Thanks, everyone. Bye. See you next week.